I wanted to share with you some work that we have recently completed, just completed as an interim step that is going to be continued over the next few years. And towards the end, you'll see why this is a work in progress and not a completed work. And the question that we're going to address is, what is the antigen that's driving not just giant cell arteritis, but large vessel vasculitis in general, if they are indeed, as we have posited in the past, part of the same spectrum of disease? And the discovery that was made by our fairly large collaborative group, and principally by Ritu Chakravarti, who has done the biochemistry for this study, is 1433 proteins appear to be antigens of some significance in large vessel vasculitis. I'd like you to focus on these two pictures here. The one on the left is a normal temporal artery. The one on the right is obviously a very altered temporal artery. There's a presence of an intense inflammatory response. There is modification of structure, and as you would guess, there's modification of function. And we know a lot about what has taken us from normal to disease and vessel destruction. We know a lot about many cytokines and growth factors that play a role in the injury and repair of injury. But what we don't know is what the trigger is. So let's review some of the pathogenesis-related work that's been done by many people in the field, but I like particularly the work that's been done by the Garanzi's lab. They were among the first to look at peripheral blood as well as temporal artery biopsies and teach us that Th17 cells and their principal cytokine, IL-17, is markedly increased in the blood, and you could see these cells and the cytokines in tissue by immunohistochemistry, as you see here and in higher power on the right. Now, if you treat a patient who has this kind of picture, the clinical picture of GCA, and this type of histopathology, treat them with high-dose steroids and come back, and then look at that biopsy again later, you see little to no signal for IL-17, but you do see a signal that is characteristic of Th1 type cells, and that's gamma interferon, which led the Garanzi lab to state that while GCA is a disease that has exquisite sensitivity in terms of the Th17 pathway, exquisite sensitivity to steroids, the Th1 pathway, and particularly the gamma interferon pathway, is not significantly affected by steroid therapy. Now, along comes, several years later, Terrier et al. In the Patrice Kakoub lab in France, again publishing in A&R in 2012, confirmation of the Garanzi findings. And here we're looking at immunohistochemistry again, interferon gamma being increased. You can see in the magnified view the immunohistochemistry identifying gamma interferon. You can see here, catty corner from that, the IL-17 signal, again by immunohistochemistry. And the other thing that this group showed us in this work was marked increased expression of IL-21. Now, they went beyond that to look at the relationship of IL-21 to Th17 and Th1 cells and showed that IL-21 correlated in direct proportion IL-21 to the presence of increase in Th17 and Th1 cells, suggesting that IL-21 is driving those two pathways. And they then went ahead and did ex vivo studies looking at the response of CD4 positive T cells to stimulation with IL-21 and demonstrated marked increase in Th1 and Th17 cells with their associated cytokines, but they also demonstrated a marked decrease in Tregs. And by immunohistochemistry here, you can see the marker FOXP3 for Tregs is barely perceptible in this biopsy specimen. So following that ex vivo stimulation study, they said, well, what happens if we repeat that study, but this time we block IL-21? And what they showed was a marked decrease in IL-17 and gamma interferon and an increase in Treg cells. And so their conclusion was that IL-21 appears to be a more proximal critical cytokine in driving Th1 and Th17 cells and in driving also the diminution of Treg cells. They thought, well, might this not be a good target for therapeutics? And that's where that stands at this point. I'm not aware of any anti-IL-21 studies that have been drafted yet. Now, ever since I heard about GCA as a fellow, and many years later when people started doing immunohistochemistry and other studies to demonstrate what cytokines were upregulated, all we talked about was dendritic cells, macrophages, and CD4 T cells. But in fact, B cells are upregulated in this disease too. So we see here from the work of the Brower lab that CD20 cells are present in temporal artery biopsies. Plasma cells are scant. And of course, as we have seen by many others demonstrated, T cells are markedly increased in temporal artery biopsies. So the question that we asked is, this is all well and good, and blocking IL-21 is a very attractive target, but really, is there something more proximal than targeting that cytokine that could give us a better sense of what is driving large vessel vasculitis from the start? What is the precipitating antigen to which this immune response is directed? And we asked this question in the study that I'll show you, not just for GCA, not just for Takayasu's, but also for idiopathic focal vasculitis. And we thought, if we were going to approach this, what might be the easiest way, although this was by no means easy, what might be the easiest way methodologically to answer this question? So even more than temporal artery biopsies, we get access to aortic specimens, thoracic aortic specimens from this center because we have this incredibly large thoracic aortic aneurysm group within cardiothoracic surgery. And they do over 700 thoracic aortic reconstructions a year. Now, when we look at thoracic aortic aneurysms nationwide, there's about 38,000 per year presenting as new findings in the United States. And 4 to 8% of these, in our center here, about 4 to 5%, but nationwide, it's estimated by some investigators that up to 8% have the finding of aortitis, not due to an infectious agent that has yet been identified, but associated with giant cell arteritis or Takayasu's, or I'll come back and explain to you what focal idiopathic aortitis is. So we said, if we could just capture those specimens, and certainly you get a larger sample of tissue than you do with temporal arteries. If we could just capture those specimens and get also consenting patients to give us autologous blood, we could begin to answer questions about, is there an antibody response to a large vessel vasculitis antigen? And that's what actually led us to this paper. So this is cutting to the chase and telling you that, yes, we did find an antigen that patients were making antibodies to, and the antigen is 1433 proteins. And that part of the work was done in the Chakravarti lab, and we couldn't have done this work. You can see there's a lot of authors here without the collaborative, collegial cooperation of our people in cardiothoracic surgery. So this was a very rewarding collaboration. Let's take a step back and look at large vessel vasculitis in general. So Alison Clifford, when she was here, she was a wonderful fellow and great investigator, and presented this work at the ACR last year. And the question that she asked is, if I look at all of the thoracic aortic aneurysm surgeries that have been done at the Cleveland Clinic over a period of about 16 years, over 7,500, averaging about 500 a year, but in more recent years, 700 a year, how many people would I identify who actually have aortitis? And she analyzed the data on the first 200, approximately 200, 196, out of 300 that were identified. So this is where that 4% figure comes from. And of those people who were among the 4% who had aortitis and thoracic aortic aneurysms, about 21% had GCA. Only 7% had Takayasus, and the vast majority had focal idiopathic aortitis. Now, if that's a new concept to you, I'll tell you that this is an area that we've been very interested in beyond the aorta. Focal vasculitis restricted to one site within one organ is not all that rare. It's been reported in almost every organ system in the body, and if the lesion, the focus, is resected, that often represents surgical cure, and the patient does not need systemic immunosuppressive therapy because the lesion is localized. It's not a systemic disease. And that is the case for how we've defined focal idiopathic aortitis. The histopathology is like GCA and Takayasus, but these patients do not have systemic features. They do not have, as best we can tell in evaluating them, a systemic disease. Their surgery was done principally for hemodynamic problems, severe aortic regurgitation, a progressively enlarging aneurysm, and they are discovered to have aortitis mostly on pathology. And everybody is then surprised that this patient was among that 4% or so who had aortitis. So these folks often, when they've had labs done before surgery, their labs have generally been normal as well. So the methods that we employed to ask our question, what is the antigen, involved getting our cardiothoracic surgeons to take from an aneurysm like this a 4 to 5 centimeter specimen, apart from that which went to routine pathology, save that for us along with autologous blood. The tissues were maintained in a sterile fashion because we're doing parallel studies on microbiome. And the tissue was then homogenized, lysed, separated so that we could then run them on gels, Western blots, and see what were the many proteins that were part of this aortic wall. And on those homogenates of aortic wall, we would then, on those gels, run the patient's serum to see if there was antibodies within the serum that was targeting a particular antigen. And then If there was binding, we ran anti-human IgG on the column to see if we could then visualize the band. And once that band was recognized, we then cut it, sent it to our mass spec core to get more specific identification of what that protein was. So I'll tell you that, yes, we did find a reactive species within these aortic specimens that was a 30 kD band, and this was being targeted, and we see this in an ELISA assay where patients with large vessel vasculitis have significantly higher titers of antibody than patients who are our controls with a variety of different diseases, and also our aortic aneurysm controls. So these are what we called match controls. These are patients who had aortic aneurysms but didn't have inflammatory aortic disease. So they could be people who had aortic aneurysm because of chronic inadequately treated hypertension associated with aging. They could be people who had bicuspid aortic valves, for which 50% have associated aortic aneurysms, or they could have a true connective tissue disease like Marfan's or Ehlers-Danlos. And these people had a low titer response to the 30 kD protein. So that 30 kD protein was then looked at in terms of the patient's aortic lysates, but also control aorta lysates. And so when we looked at the first 15 patients in this way, we saw that patients with large vessel vasculitis in 14 out of 15 cases, not here for number 3, but in 14 out of 15 cases, were reacting to this 30 kD protein, whether it came from the aortitis sample or came from the control aortic aneurysm sample, the bicuspid aortic valves, the Marfan's, the Loeys-Dietz patients. But when we looked at those match controls and put their sera on these gels, there was no reactivity. When we looked at other controls, and they're more thoroughly delineated down here with ELISA testing, you can see that there were very infrequent positives, and 18 out of 23 patients with large vessel vasculitis reacted to this 30 kD protein. So there's a caution here. The numbers of controls are relatively small, and this work has to be confirmed in a much larger cohort, including the control cohort. But given that limitation, we're still looking at 78% sensitivity and 94% specificity. Now, when we subjected the 30 kD protein to mass spec, that is, looking at this protein in terms of its components with ionization and trafficking in an electromagnetic field and mass to charge qualities, we find that our patients were making antibodies, whether they came from control samples, C1 and C2, or large vessel vasculitis aortic samples, L1 and L2. By mass spec, we identified all of the 7 isoforms of 1433. But our patients appeared to be making antibodies only to the eta and zeta isoforms. So I didn't know anything about 1433 when Ritu came to me and said, oh, our patients are making antibodies to 1433. So what the heck is that? And what I subsequently found out over the next 3 years is I should have known something about this protein, this family of proteins, because they're ubiquitous. They're in every nucleated cell in our body. They're in the plant kingdom as well as the animal kingdom. And they have 7 isoforms, and then there are subsets of those isoforms. They function as dimers. They are sterically arranged as dimers. and those can be heterodimers or homodimers. And people who have been looking and studying at 1433 tell us that they actually are involved in 200 to 300 different metabolic and immunoregulatory pathways. So they're critical to normal cell biology, and I'm amazed that I knew nothing about it until the tender age that I am now. And so how do they work? They work by being part of what some people call the interactome. And by that, they're meaning that they facilitate or they inhibit protein-protein interactions. And they do that in an isoform-specific manner. So what are some of the processes that are regulated by 1433 in terms of protein binding and subsequent modification of those proteins in a way that affects their function? Well, many pathways that are involved in cell cycle control, signal transduction, DNA transcription, protein trafficking, apoptosis, isotype switching, many different processes. And they do this by the 1433 binding to serine or threonine phosphorylated proteins. And when they bind, what happens to those proteins is modification of their conformation, their stability, their localization within the cell, and their enzymatic activity, just to mention a few. So if these proteins are present in every nucleated cell in the body, not red blood cells, you'd expect that in disease states, their concentrations might be increased. And so they are in rheumatoid arthritis, neurodegenerative disease, many different forms of cancer, infectious diseases, in post-op states where there's tissue damage, remodeling, and healing. But the interesting part of this is that some people make antibodies to 1433. It's not just part of the story that I'm telling you about our own work. People make antibodies to 1433, and in some cases, that 1433 has been modified. Modified through post-translational modification in some cases. For example, in rheumatoid arthritis, 1433 is citrullonated. In breast cancer, 1433 sigma is hypermethylated. In many other diseases, there's an immune response, but we don't know whether or not the 1433 has been modified in some way or if it's native 1433. So back to where we were a few slides ago, the large vessel vasculitis story. So I've told you that in large vessel vasculitis, 1433 is a target for antibodies. We next wanted to be more certain that this was a real observation about especially epsilon and sigma. So we looked at reactivity on Western blots. And here we see commercially available antigen that you can buy, and epsilon, zeta, and here gamma, because there was a slight on mass spec, there was a slight signal for gamma as well. And when we reacted patient sera with that, we confirmed that yes, there was binding to epsilon, zeta, and gamma. There was no reactivity with control sera. And as a quality control for this, we had purchased specific antibody, specific for this purified purchased antigen or group of antigens. And you can see that, in fact, the antigen remains intact. There is immunologic reactivity that can be demonstrated for each of the antigens. And when we applied by immunohistochemistry a commercial antibody to see whether or not there was reactivity in tissue that was beyond background 1433, since 1433 is present in all normal nucleated cells, but not to a degree where you can see it here within the unaffected, non-inflamed tissue. But here you can see there is antibody, this commercial antibody targeting 1433 in tissue, but we see the same pattern in an adjacent cut of this specimen with patient sera. So we're confident that there is an antibody and it is targeting antigen within these temporal arteries. So our conclusions are that in large vessel vasculitis, and I started out saying this was principally a discussion about GCA, but our aortas are not just GCA aortas. They're Takayasu's aortas, and they're focal idiopathic aortitis aortas, and we see this kind of reactivity in all of them. Again, enhancing our argument that these diseases may be part of a spectrum and not all that separate. Now, as I emphasized before, it's not just 1433 in general, it's the epsilon and zeta that appear to be the reactive species. And we also showed that 1433 co-localizes with vascular smooth muscle cells, which are those cells that appear to be most intensely targeted when you look at histopathology of large vessel vasculitis. So what's next? What are we going to do in the future to build on this? Well, certainly we need a larger cohort of patients. We need to test this against hundreds of patients who are patients with vasculitis who don't have large vessel vasculitis. We need to test it against hundreds of patients who have a variety of autoimmune diseases and other diseases. And we need to see if this holds up in this larger cohort. We need to get a better understanding about whether 1433 here, as in other studies and other diseases, is a modified antigen. Has it been modified in a way through post-translational modification or mutations so that this is no longer self? And in not being self, there is what you'd expect to see, a normal immune response to a foreign antigen. Or is it, I think, less likely, loss of tolerance to this antigen? And we don't know, but we hope to find out. We need to know, as Lenny implied in the beginning, will this be, in this larger cohort that we'll study, will this be a compelling diagnostic test for large vessel vasculitis? So that 38,000 people who present with thoracic aortic aneurysms throughout the United States and have only hemodynamic problems, can we identify some of those people prior to their surgery as having an inflammatory condition? And I don't know whether or not that will affect their management, but it seems like if we have an opportunity to know which aneurysm is inflamed and which is not, that may provide some therapeutic leverage. Certainly, it'll change our approach to going back and talking to that person and ferreting out other symptoms. And then, if this turns out to be really quite specific in a larger cohort for large vessel vasculitis, will we be able to use titers of antibody or the protein itself as a surrogate marker of disease activity? Because sedimentation rates, CRP, they're pretty good, but they're pretty nonspecific as well. And then we can't lose sight of the fact that we did the easier study. We looked for antibody in patients' sera to a putative antigen that might be important in pathogenesis. But we still know that this is a lesion in each of these diseases that are driven primarily by T cells, Th1, Th17 T cells. We need to determine whether or not there's T cell reactivity to these antigens. And then if there is, the Holy Grail would be, if this is really playing a role, do we have opportunities to block immunologic reaction to 1433? Will that offer us therapeutic opportunities? So that's where we are now. This is where we'd like to go in the future with some of these questions. And we have much more work to do down the road than the work that we've already done. Thank you.